So my name's Ian, by the way.
And I'm Maria. Can you all hear me?
Yes.
Okay.
And so we are presenting on a paper titled The Mechanical Rigidity of the Extracellular Matrix Regulates the Structure, Motility, and Proliferation of Glioma Cells. By the way, can you guys see the little panel that has all your faces in it on the side? Because I'm trying to minimize.
No.
Okay, perfect. Okay, so we don't have too much of a super personal connection, but we really enjoyed the week 5 paper. And that paper talked a lot about ECM rigidity. And I think a lot of this stuff that you see in this one will probably remind you guys of that one, if you guys remember it at all. But even if you don't, it still might remind you guys a little bit. There's a lot of similarities between the two. So okay.
So our paper mostly focuses on glioblastoma multiforme, or GBM, which is an aggressive brain cancer that originates in Galal cells. So the disease progresses pretty rapidly due to tumor cells diffusing into the brain parenchyma, which I'm not sure how to pronounce, or other surrounding tissues. So this kind of diffusion or migration depends on 3 things, and that's actomyosin-generated contractile forces, changes to the cytoskeleton, and cell matrix adhesion complexes. And then these factors, along with previous research that showed that GBM tumors are found to be stiffer than surrounding cells, led to the hypothesis of the paper that mechanical cues from the ECM influence growth and invasion properties of GBM tumor cells.
So basically, multiple glioma cell lines were cultured on fibronectin-coated ECM substrates of various rigidities, spanning between normal and tumor tissue as far as those rigidities go. So for values, the sort of softest substrate will be 0.08 kilopascals. So again, 0.08. The kind of middle sort of rigidity will be 0.8 kilopascals. And then the highest rigidity that they're going to use that you're going to see a lot is going to be 119 kilopascals, which is very rigid. You're also going to see in some of these figures, they're going to...
I just want to ask. So can you just repeat those numbers again for me?
Yes. So, those are in other slides. So that's why I didn't write them down, but I'll try and get you guys.
No, you're good. You're good. I just was curious. Could you just repeat them for me again? I just want to make sure I got it right.
Yeah. So the softest substrate will be 0.08 kilopascals. The kind of medium rigidity substrate will be 0.8 kilopascals. And then the most rigid will be 119 kilopascals.
So what's up with the range there? That's crazy. So it goes from 0.08 to 0.8 to 119.
Yeah, I think I think the point there was basically that 0.08 is the softest, and that value makes sense, and that's fine. 0.8 is kind of in the middle, and is more realistic. I think 119 is kind of a ridiculous jump, but I think that is so that they could kind of exaggerate and get kind of very clear data. They're also going to use, one thing you're going to see in this paper also is they'll use glass. And glass, when glass is the substrate, the ECM substrate, they're putting these cells on, culturing these cells on. Glass is going to be synonymous with about 100 to 119 kilopascals. So it'll be when you see glass, just think of the basically the highest possible rigidity as well, or something very close to it. And so the goal.
I have a question. Where does the normal and then tumor rigidity lie on that scale?
So they don't actually say that. That's just kind of, that's normal, and tumor tissue is kind of just something they say at one point in the paper. I believe though, based on like reading the paper and obviously in the methods and materials and stuff, I think that normal is going to be about 0.8. And then I think tumor tissue is going to be something above that. They don't really say, but that would be probably not up to 119 kilopascals. But they actually don't really say that. They just kind of use normal and tumor tissue very loosely at one point in the manuscript.
Okay.
Does anybody we could use our brains a little bit. Does anybody remember the ranges for the soft to stiff for the like neuron to bone paper. We've got 20 of us here in this room. I think it was like four, was it like something like 4 or something for the really like neural soft tissues? And then like 40? Am I off?
I remember the number like 37, I think, for some.
Yeah,
Yeah.
35 for bone. And it was like 4, I want to say it was like 4.4 for like neuron. Does anybody kind of remember what the muscle was? Maybe like 20. I don't know. Okay, 35 for bone. So bone is probably stiffer than like the starting of bone is probably stiffer than a tumor. So tumor-ish is probably, I would maybe say like 10-20, maybe.
Yeah, I mean that would make sense, but again, they actually oddly enough didn't really specify that.
Yeah.
So, anyways, so the goal of the paper is to kind of analyze these ECM mechanical cues and what effect they have on the GBM tumor cells, as far as really 3 criteria. The first is kind of confusing. They say spread, or they use kind of the words spread, migration, and motility pretty interchangeably, but that'll be the first criteria. The second is growth, and the third is proliferation. And then, interestingly enough, one thing they do that to us kind of completes the data set of this paper that I really liked was that they start inhibiting non-muscle myosin 2 and its upstream regulator ROCK, and then seeing if these cells are still sensitive to the ECM mechanical cues. So does this sensitivity to ECM cues still exist, and is that still shown with these cells after non-muscle myosin 2 and its regulator ROCK are inhibited?
So for the first figure, they wanted to see how glial cells would change when grown on different ECM rigidities. And they found that changes in ECM rigidity do change cell morphology and cytoskeletal organization. So for A, they stained F-actin green and saw that F-actin increased as rigidity increased. They also saw on softer substrates, so again, that 0.08 or 0.8, they had smaller spreading areas and also tended to be a little bit more round, which you can also see in B and C towards the right. More specifically in B, they stained F-actin green again and vinculin red, which is a focal adhesion protein, if you don't remember, and saw that both increased as rigidity increased. And then C just kind of showed the vinculin signal alone. And again, it showed that it increased.
Yeah, so for figure 2,
Sorry, can you just go back? I just wanted to look at that picture a little bit, just a little bit longer. Thank you. Yeah, that's all. This is all pretty nice, right? This all kind of checks out with what we know. Okay, cool.
Yeah, this is pretty good stuff.
Yeah, cool. Thank you.
Yeah, so this next figure basically is looking at the effect of ECM rigidities on motility. So again, motility is somewhat synonymous with like movement, spread, migration, I guess, or similar sort of synonymous terms. And they do use them pretty interchangeably in this paper. But for A and B, they're looking at basically the same figure for A and B, looking at the exact same thing. They're just looking at 2 different glioma cell lines. So for A and B, you see you both see the same thing in both those instances, which is that the more rigid these substrates are, the more that these cells are migrating and the more motility that they have, which they measure with average cell speed. And so you see as you go along there, again, more rigidity is leading to more migration and motility for A and B. And C gives you kind of just a look at that so you can visualize it with a time course. Again, like I said before, glass is going to be synonymous with just a very, very rigid substrate, very similar to the 119 kilopascals. So what we're seeing in the first row in figure C, those are the cells cultured on glass. We're seeing smooth and quick migration. They don't really explain in the paper how to kind of look at this and visualize this. They kind of just try and explain it themselves. And what they see here is smooth and quick migration, which I guess when you compare it to that bottom figure, the bottom row, which I'll talk about, you can kind of see some of the differences. But for the kind of medium value, the zero 0.8 kilopascals, what they're seeing is what they call stick slip migration. And so they're seeing migration and movement still, but it's in this format that they call stick slip. And stick slip migration is essentially when the cell thins itself, extends towards the leading edge, and then the trailing edge kind of just snaps forward. And you can see kind of how that between the 90-minute and 92-minute mark for that middle row, you can kind of see that trailing edge snap forward. And so this would be something that it'd be kind of cool to see like a movie of, I feel like. But actually from these images, you can kind of get the gist of what that stick slip migration looks like. But again, it involves the cell sort of thinning itself out and extending towards that leading edge before that trailing edge snaps forward. So it is kind of a cool thing to look at. But basically, they're showing that migration is still occurring at this particular substrate rigidity, but it's not as smooth, efficient, or quick as you're seeing with glass. And then finally, with the 0.08 kilopascals in that bottom row, you're just seeing basically no migration. The cells really, they failed to migrate. They really weren't able to get anything going on that softest substrate.
Hi. This is that you did a good job there. This is really cool. I just want to add, I kind of want to add in a little discussion question, if that's okay. Can I do that?
Mm-hmm.
So I'm looking at this glass migration style and like that stick slip that Ian described, and you could see those, you know, he described those differences so nicely. And in glass, the migration, those sort of the orientation of the cells and their shape and the way they look very much like these like keratocytes or keratinocytes that crawl around, and they're very good migrators. And you could see that they have this huge like fan-like lamellipodia up at the front, which is full of like that branched actin and all that. And you could just see how big it is compared to the one at 0.8 kPa. And so then I wonder if that is affecting, so since you have that huge fan out there, you have all that membrane spread out, right? A huge amount of membrane. And so then these cells wouldn't be able to form these like sticky tails because the membrane would be under higher tension. Does that make sense?
No, yeah, that I mean, that makes a lot of sense to me. And does anyone else have any comments on any of that as well? Okay.
So then I guess my question would be is there a way that we could, let's say, using things we know from this class, kind of design a readout to maybe look at membrane tension in these two cells and see if they're different in a cell that's on, like in that back end part, in that tail part? How could we maybe have an experimental setup that would allow us to observe changes in membrane tension? Any ideas?
I don't know how this might work by using specific tension or specific rigidity of substrate, but there was the one traction fluorescence imaging, I think it was called.
Yeah. So that would be looking, but that would be looking right at the focal adhesions. So we want to, right, but that would look at tension, but we would probably have that figure in the paper already. And now we want to look at like just the membrane. And can we do that using anything that we know about? Do we have any tricks that we remember? So I was thinking maybe, what if we looked at calcium imaging and we used piezo channel blockers or not? So piezo opens with membrane tension, right? So if these have piezo, or we could overexpress piezo, then we could do calcium fluorescence imaging and see how much those channels are being opened in those tails by measuring the intensity of a calcium probe that is labeled with GFP or whatever. Does that make sense?
Yeah.
Cool. So, sorry, that was just like a, you know, if I was like watching you give your science talk, and then I was like, ooh, cool. This is like a cool science question that I would want to try to explore. And we kind of already learned about that, so I just wanted to bring it back. Sorry, I will shut up again.
Yeah, like I said, that's one thing we like a lot about this paper, and kind of our connection to it is, you know, we've gone over some of this stuff before, and I think week 5 was a pretty good example. So yeah, but anyways, I think, you know, I think kind of like a little bit like what Tori was saying, I think one of the coolest things, you know, with this figure in the paper, but also with this figure in particular, is just, you can kind of see, especially if you look at it for a little while, some of the differences between these cells, like the big fan for the ones on glass. And then I think that stick slip thing between 90 and 92 minutes is really cool, how that back edge just kind of all of a sudden snaps forward, I think is pretty cool. But so yeah, so if no more questions on this, figure 3.
So for the third figure, they wanted to see if Galal cells would proliferate more or less on different substrate rigidities. So for this experiment, they used 2 different Galal cell lines, which is seen by A and B, and they used a BRDU assay, which is where they introduced the chemical BRDU to cell cultures, and it's incorporated in replicating DNA. They then introduce an antibody that detects this chemical, so they can determine which cells are proliferating, because obviously proliferating cells replicate their DNA. They essentially found that with both cell lines that there was more proliferation as rigidity increased.
Oh, Tori, did you want to, or, you know, just in case anyone want to see this figure again, it's pretty simple, but all right.
I got it. I got it. Thank you.
Okay, cool. So yeah, so this 4th figure is kind of a lot. Admittedly, it is a bit hard to understand and explain, but I think we got a pretty good grip on it, especially because I think figure B really goes into detail, some specific detail on kind of the takeaway of figure A. It looks at it a lot closer. But what they're looking at with figure A is basically cell morphology and migration patterns with different inhibitors, which you see on the left, on the side of the figure, and at different stiffnesses, which you see at the top. And what they're asking is, are glioma cells still sensitive to ECM rigidity in the presence of these different inhibitors? And so blebistatin, which you'll see on the left there, that's going to be an inhibitor of non-muscle myosin 2. That Y27632, that is an inhibitor of ROC. And then cytochalasin D is an inhibitor of actin polymerization. And then at the top there, you have those different substrate rigidities, ranging from the glass, which is very stiff, kind of that, you know, I know that I said that it's somewhere around the 119 kilopascals. I'd say as far as results are, it is, but I guess like in this figure, they're kind of asserting that glass is just like astronomically a higher stiffness than the 119 kilopascals, but beyond 119, the data is pretty much all the same, and you can see that a little bit with these panels as well. But so that's what they're asking is, are the cells still sensitive to ECM rigidity with these inhibitors? And what they found was that when they inhibited ROC and non-muscle myosin 2, those middle rows, what they found was that basically when they inhibited those 2 things, that blunted the sensitivity of these cells to ECM rigidity. So these cells no longer, you know, had morphology or migrational patterns similar to what you'd see on these typical substrate rigidities. So when they knocked out ROC and non-muscle myosin 2, that's when the sensitivity to rigidity was diminished. But interestingly enough, and they explain this with figure B, they take a closer look at this, when they inhibited non-muscle myosin 2 or ROC for these softer substrates, that actually not only blunted the sensitivity to rigidity, but it actually rescued the ability of these cells to migrate and to show certain morphology to an extent that they wouldn't be able to if they're on a soft substrate and if these components were not inhibited. So for figure B, they take a closer look at this. This is again, so figure B is the cells on the softest substrate, 0.08 kilopascals, with ROC being inhibited. And what they're showing here in this time course is that after a while, by inhibiting ROC, the migration ability and the stiffness dependence was eliminated, and migration ability was actually restored. And you can see that a little bit in that time course. So basically, what that was kind of their big finding here was that by inhibiting ROC or non-muscle myosin 2, these cells didn't depend so much on these ECM rigidity cues. And then what they found with that last inhibitor, cytokalasin D, the inhibitor of actin polymerization, it's basically just kind of another control, because the cytoskeletons collapsed for all cells on all substrates when they use this particular inhibitor. So does that kind of make sense to everyone? Was there anything I kind of missed out on there?
No, it was good. I think it's interesting that the cells change when we hit them with these inhibitors. They kind of lose, like, they especially in the blebbistatin one, like they look like they're spreading out and crawling around. It's so interesting. Like you take away their mechanical machinery, and then they look more like the stiff cells. Does that make sense?
Mm-hmm.
I think that's I would have kind of expected the opposite. I would have expected them all to look like little rounded up kind of weird guys, but yeah, they're an interesting little cell type. So yeah. I think
and
that's
then
cool.
there was one more other thing I was going to mention as well. Yeah, I forget exactly what that was, but basically that's this figure. And there's actually a theory. So one of our discussion questions is about why that might be, and there is a theory that the authors kind of put forth on that, and so we can talk about that at the end, but we will talk about that. But as far as conclusions go, basically overall takeaways. Higher ECM stiffness leads to more cell spread and migration. It enhances the expression of contractile proteins. A stiffer ECM equals more cell proliferation. And then again, like I talked about that last figure there, perhaps the most interesting takeaway is that inhibiting non-muscle myosin 2 or ROC diminished sensitivity of glioma cells to ECM rigidity. And one thing that they say as well is that non-muscle myosin 2 and its activators are a critical component of the ECM rigidity sensing pathway. So it kind of makes sense a little bit as to why that might be. But there's actually kind of a deeper sort of theory, or whatever you might want to call it, that the authors talk about as far as like specifically why certain cells can still exhibit some migrational patterns even on soft substrates, even with non-muscle myosin 2 or ROC being inhibited.
So here's the model we drew to just kind of sum up most of the results. So as you go more towards the right, the ECM gets stiffer. And then we kind of tried to portray that the cells proliferated more by putting more cells towards the right, and then also that they got larger as they had bigger areas. We also kind of tried to show with an arrow at the bottom that they do get faster as it gets more rigid. And then lastly, wanted to show that the cytoskeleton rearranged as the substrates got more rigid. So we tried to add more F-actin and vinculin on the cells on the right. The cells on the right, yeah.
This is very nice. They are also very cute. They look like little one-eyed eggs. I like them. But yeah, no, this is great. Thank you.
Yeah, so as far as this next stuff, so before we get into the discussion,
I actually have a question for the model here. On the left side, we have this like separation of actin and vinculin. Was that actually? I don't remember if that was actually
It was not
true.
intentional, no.
Oh, okay.
Sorry.
No, it's all good. It's all good. I was just curious. It's okay.
Okay, so then as far as the discussion and all that stuff goes, before that we have a quick analogy. I just kind of scrapped this together, did my best, but I figured that the glioma cells with rigid substrates are like the bars in Ann Arbor before the coronavirus stuff. Basically, a lot of movement, a lot of activity, a lot of proliferation, a lot of bodies, like cells. Basically, if each cell is a body, there's just a lot of activity and a lot going on. And then the glioma cells with the soft substrate are the bars during coronavirus. I guess not anymore. They're not letting anyone in, but perhaps even after. Basically, no movement, no activity, not a lot of bodies, not a lot of proliferation, or just not much going on. They can't exactly thrive under those conditions. So yeah, that's what I got as far as the analogy, and then yeah, we got a couple discussion questions as well.
Yeah, so we have discussion questions for either just the class or maybe breakout rooms. So why might inhibiting ROCK or non-muscle myosin 2 diminish sensitivity to ECM rigidity? And also, what are some potential clinical applications of this work? In other words, how can the results of this paper lead to possible therapies or better diagnostic tests to improve the prognosis? Because currently, if someone gets diagnosed with it, the prognosis is like 15 months before they might die, so it's not great.
Oh, great. So I'll open up all the rooms here in a second. Let's just go over the questions again. Okay, so if we inhibit myosin through ROCK or myosin itself, why does that affect cell sensitivity to the ECM, and what are some potential clinical applications of this work? Great questions.
Also, that second one, we acknowledge that second question is pretty darn tough. So obviously, we're not really expecting anyone to have any incredible like answers right off the bat or anything. So apologies, that is a tough one.
No, I think those are always great questions, because anytime you like sit down and write about science, you always got to come up with this stuff. So these are good questions to just kind of prime our brains with. So I'll open them up, and then we'll come back. So go ahead and join up, and we'll spend about a minute or two.
Wait, Tori, should I still share my screen, or should I just not?
Yeah, you can just leave it up, because when they go in their breakout rooms, it doesn't. Gotcha. They don't even see that, but we're going to come back to it. So you can just stay in this room, and Maria, you can stay in this room as well. You don't have to go out. I think people should be good without you. Oh, Maria actually left. It's all good though. Just gotta give them all the answers. This is always a fun thing, because I always know like when people are AFK because they don't join their breakout room. It's like, okay, you're in the bathroom.
Yeah, I'll give him credit. You almost got me. What was that, yesterday? I was like, I got back from the bathroom right as I heard you guys talking about like, oh, you might not be here. I was like, fuck no, I'm here. I mean,
also I understand that like, people got lives, and this whole time of life is crazy. So some of us might just have our computer on and be doing other things, because that's what we got to do. So, but
no, of course.
I totally get that. A lot of us are here and able to do it, so it's great. All right, I'm gonna close these up. Are you ready for everyone to come back?
Yeah, totally.
Cool. Welcome back. Or if you never left, thanks for staying.
All right, so do we want to talk about that first question then? anybody has any potential answers or anything, just or any thoughts, even anything they want to bounce around.
I mean, I thought it was so interesting that when you inhibited ROC or myosin, that the cells looked like they were on the stiff substrate. That's so weird.
That was pretty cool.
I don't really know. I don't have a mechanism in my brain for how that works.
I have kind of like a light sort of answer that the authors talk about that they don't go into like super big detail about, because obviously I'm sure this is kind of their next steps is to figure that out. But I do have a theory that they talk about. So
let's see if anybody else has something.
That's what I'm saying. Yeah. So if we can just even get any theories and what people might think, I'm sure nothing will sound too crazy.
I mean, we kind of just talked about how rock and myosin is how the cell like kind of responds to that rigidity, like in a way. So like if you're inhibiting that pathway, like they can't really like respond to the rigidity in the same way.
Yeah, so you absolutely have that first part correct. And no, yeah, you have that first part correct. Does anyone know what they think might happen? Like as far as it's kind of a two-part answer. So you got the first part. This is even what you said is even obviously what the authors said. Does anyone have an idea what the second part of the answer might be? What happens once that non-muscle myosin 2 based sort of restrictions, once those are lifted? Does anyone have any ideas?
What do you mean by once they're lifted?
Or essentially once the cell spreading, et cetera, is no longer non-muscle myosin 2 based. So once non-muscle myosin 2 is inhibited, does anyone have any idea what might happen in the absence of the non-muscle myosin 2 based?
I have a thought. So the way the cells sense rigidity in a nutshell is that it pulls at the substrate and sees how much it moves, right? Mm-hmm. Well, if it pulls at a stiff substrate and doesn't move, and the substrate doesn't really move, it doesn't shift in response, then it thinks, okay, so I'm on a really hard substrate. Mm. Well, if it thinks it's pulling and it doesn't move, then it probably thinks the substrate is very
that makes sense to me.
Stiff. So honestly,
that Aaron, makes sense.
You pretty much got it. The only thing I'll add to that, and again, this is a very small mention that the authors make in the paper. I don't expect really anyone to remember this, but you pretty much got it. The only other thing is the authors posit this idea that perhaps in the absence of the non-muscle myosin 2 based sensitivity, basically they think that a RAC mediated cell motility is basically coming into play. So they think some sort of RAC mediated cell motility pathway or machinery, they think that that's what might be enhanced and might be taking over.
Yeah, that makes sense because you're inhibiting Rho, and they're like counter. They inhibit each other, Rho and RAC. So then you have up RAC, you get more spreading out, and then also what Aaron is that also just like force regimen kind of things that we've learned about, and what he talked about is also happening. Yeah, that totally makes sense. Cool. Yeah. Thanks for teaching me that, everyone.
And then for that second question, do we have any, again, just rough ideas or thoughts, or really just comments, even just whatever you guys might be thinking right now? Well, all right.
So let's just go ahead and let's move on. We have spent quite a bit of time here, so let's try to.
Oh shit! Yeah, it's been a while.
Can we wrap this up?
Yeah. All right, guys. So, anyways, if anyone else has any final questions or anything or thoughts, then whatever. Maria, if you want to spend like a minute talking about clinical stuff, I don't know if we have time for that, so we might just want to move on.
No, we definitely have time for that. Yeah, Maria, if you would like to.
Oh yeah, you have any thoughts on that, Maria?
Yeah, so we kind of just talked about like interfering with mechanical signaling or mechanical remodeling so that the GBM cell motility or invasion could possibly like slow down or stop. It's kind of what the paper talked about. So that's
Yeah.
kind of the ideas that we were throwing around, but we don't really know other than that.
Yeah.
One idea that popped into my head was like, like, I don't know, trying to really affect the mechanical environment of tumors and the surrounding tissue even of tumors. And how could we like locally deliver, like ECM degrading enzymes and things into tumors if we, to change their rigidity or add extra ECM depending on around the tissue to keep the cells in the tumor, something like that, like, affecting the mechanical landscape of tumors.
Good idea.
All right. So
I like whisper, good idea.
all right. So I'll stop sharing the screen then. Thanks guys for listening. And yeah, let me reach out to us if you have any more questions or anything like that.